Thank you so much, Dr. Holland, and thank you for hanging in there. I might try to go a little bit fast because I know that we're over time. The title of my talk is a little bit misleading because anybody who knows me knows that I'm actually all germ cells all the time, and to the extent I do worry about fetal and early life vulnerabilities, I'm mostly thinking about what's going on inside those little gonads. So with that caveat, here's an overview of how at least I conceptualize how we think about risk over the lifespan. And actually, Dr. Bishop sort of introduced this concept just now in that question, with a concern that we tend to focus on the somatic risks, and we do tend to oversee some of the risks that might occur during gametogenesis and through the life of the germ cell. So if you see here, the lifespan, as you guys know, the life cycle starts long before conception. That's not in the conventional wisdom, but you're all scientists and you know that our lifespan actually, or life cycle starts in the primordial germ cells inside of our parents. So in some ways, although I was born in 1965, I was really born in 1930 and 1937, which were when my parents were gestating. From my point of view as a non-scientist and somebody who works as an advocate for an exposure paradigm, what I see is toxicology very, very much focusing on the very last part of the human life cycle, right? Here's the construction guy over here on the right, and very, very little on the preconception germline exposures. Now, there's been some positive development in the sense that we're looking more at fetal effects in terms of DOHAD, and of course, the field of teratology, which really focuses on gross anatomical defects. That's been around for quite a long time. But we really are way far behind where we should be in terms of thinking about the toxicological impacts on the germ cells. Now, my primary concern with the germ cells is in the very, very early stages. You guys have probably seen versions of this slide maybe a half dozen times already at this conference, and I won't go over it. But for a variety of reasons, there are multiple layers of vulnerability in the early germ cell. And I feel kind of stupid talking to this audience about it because you understand this stuff better than I do. But these include methylation, the rapid demethylation, and of course the remethylation in a sex-specific way, and the genomic imprinting, chromatin reorganization, histone modifications, the swapping out of histones for that happens later actually, the swapping out for the men, but the change in the transcription patterns, X chromosome reactivation, which I don't understand at all. There are even some other factors that may come into play, mitochondrial effects. And for persistent effects across generations, there are concerns that there is no erasure of the hominid-specific transposable elements in the germline. But long story short, I'm not a molecular biologist, and you can tell that. But long story short, these things can ultimately affect the transcriptional regulation in the offspring. But do germline exposures actually matter for offspring health? Well, I could have chosen a number of different exposures to talk about today, including things like chemotherapeutic agents or HDAC inhibitors, but for our purposes, we're just going to look at three: synthetic steroid hormone drugs, tobacco smoke, and general anesthesia. The first two, mostly concerned about fetal germ cell exposures, because as far as I know, babies aren't given a lot of synthetic steroid hormones, although they may get corticosteroids. Tobacco, again, maternal smoking. General anesthesia, that would start in utero and later in childhood. So synthetic steroid hormones, let's start there. Now, as Dr. Holland said, I'm not a scientist. I'm an ex-lawyer. Why am I here? I'm here for just a really bizarre reason. I have no long-standing interest in epigenetics, genetics, developmental biology, reproductive biology. It's just that I'm a normal mom in the suburbs of the San Francisco Bay Area, and I have 3 kids. and 2 of them have severe idiopathic forms of autism. They are nonverbal, they cannot read, they cannot talk, they cannot write, they cannot dress themselves, they cannot bathe themselves, they cannot engage in any type of conversation. They've never played with a toy. They are lacking any capacity for abstract thought or reasoning. So a very catastrophic level of disability. And I come from a family, and my husband too, where there's no disability whatsoever up our family trees. And I had no risk factors in my pregnancies at all. Very, very healthy, APGAR scores of 10, well nourished. I don't smoke, never drank, nothing. So to have this sort of catastrophic outcome come from out of the blue, nowhere, was shocking, as you can imagine, to us. But this is also something that we're seeing, and I'll talk about this later in the talk, that we're seeing across the population now. More and more of these children with unexplained autism and neurodevelopmental defects. Now, the first time I thought maybe I had a reason, I could perceive a possible reason for this, was roughly 9 years ago when, through a very serendipitous sequence of events, which I won't get into now but are described in the journal Environmental Epigenetics in this particular article, I discovered that when I was in utero in 1965 that I had been exposed to very, very heavy doses of synthetic steroid hormone drugs that were intended at the time for the prevention of miscarriage. So my mother was given these drugs when she was pregnant with me because her doctor thought that because she had 2 prior miscarriages, perhaps she was at risk for miscarrying fetal meat. And these were mostly progestins, 2 different progestins, estrogens, and throughout the first trimester, prednisolone. But did I see this pattern in other families? And when I asked around, some of the autism families that I knew, I found other families with this story. Here's my friend who lives, right near me in San Jose, the JH family. She had 5 children. 3 of them have autism. They're higher functioning than my children, but nevertheless, they do have forms of autism. She, as it turned out, just crazily enough, was born 1 month before me in the same hospital as I was in Los Angeles, and her mother had also been exposed or had been given anti-miscarriage drugs. She doesn't have the detailed records that I do, but she knew that much. Again, no risk factors up her family tree, nothing, no risk factors in pregnancy, but 3 out of 5 kids. Here's just another family that I know in the Bay Area. Both of the parents had been exposed to DES in utero, and their only son has autism. Now, I don't have time to go through all of them. I'm just giving you samples. Okay, let's do another case study. Let's go to the second form of exposure, tobacco smoke. Now, I have so many here, I could take hours talking about them, but here are just 2. It's a family where both parents had been exposed in utero to tobacco smoke. They have 3 children, 2 boys who are twins and a girl. They're heterozygous twins. And both of the boys have severe forms of autism. The girl is typically developing. And here is another family where the mom had been exposed in utero to tobacco smoke. She has a child with autism and another boy with ADHD. A very typical pattern that I see, where the girl's unaffected and the boys are kind of variably affected. And when I ask the family if there's anything up their family tree, any autism, the answer is almost always no. General anesthesia. So here are just 3 examples of a pattern that I saw very, very often among autism families. On the left, you have a mom who had 2 surgeries. She was born in Nicaragua. She had 2 surgeries as a neonate, one for removal of a tumor, one for hernia repair. She has 2 boys who are not twins, who both have idiopathic autism. Again, perfectly healthy parents, no risk factors whatsoever, no mental disabilities up their family trees, just that very early set of surgeries. The middle mother had surgery at 2 days old for removal of her adenoids. She has 3 boys. 2 of her boys have autism. One is high functioning and one is very, very severe. And then on the right, you see just a case where I'm just showing that there's also, this is with men as well, that he was exposed in infancy, again, for a hernia repair. They have one child who has unexplained severe autism. Now, of course, these are completely anecdotes, and you're like, well, gosh, you know, I'm sure there are lots of people at anesthesia have totally normal kids, and of course that's true. But my point here is that when I do ask about other siblings who didn't have this exposure, and if those siblings had, you know, autistic children, the answer is almost always no. So it's a little mini control group that I used to sort of reality check what I thought that I was seeing. Now here's another general anesthesia exposure window that came up unexpectedly. These are two friends of mine, again in the Bay Area. They both have sons with extremely severe forms of autism. And those of you who live in the laboratory world might not really know how severe autism can be. But here is my friend's son, Pablito. And that's just her trying to feed him breakfast one typical morning. He obviously has no language, he's severely cognitively impaired, and he's engaged in persistent self-injurious behaviors. My other friend on the right, her son is similar, not quite as severe. But the point is this: in conversations with these friends over the years, I discovered, to my surprise, that both of their husbands had suffered gunshot wounds in their childhood. For my friend Pablito's mom, or Pablito's dad, he was shot at age 9 in the eye and had 5 surgeries to correct that damage. My other friend's husband had a gunshot wound to the leg, a hunting accident, and had 4 surgeries at the age of 16. So I don't know very many people who've been shot, but the 2 people I know who've been shot both have, and multiple surgeries, had sons with these severe incapacitating disabilities. So this is a bunch of anecdotes. You scientists hate anecdotes, and I totally understand that. But is there empirical evidence to suggest that some of these exposures might have these effects? So when it comes to synthetic steroids, diethylstilbestrol is the best studied of all of them. There have been human studies that have looked at what happens in the offspring when there has been a germline exposure, in this case a fetal germline exposure. And quickly in males, genital tract defects, in females, menstrual irregularities, possible infertility, ovarian cancer, preterm birth, possibly ectopic pregnancy. And then published just last year in JAMA Pediatrics was a study that came out of Harvard showing an association between DES in grandmothers and risk for ADHD in the grand offspring. And then in the mammal studies as well, they show risks for cancers and neurogenital abnormalities. Other synthetic steroids have been studied as to germline effects in mammals, but they're almost impossible to do in humans because we lack the appropriate cohorts. Synthetic estrogens we've seen increased risk of cancers and reproductive tract abnormalities in the grand offspring. In the corticosteroids, mostly it's looked at behavioral and neurodevelopmental functioning, and we've seen impairments there. And then also for the environmental endocrine disrupting compounds, there's so many examples. I mean, I could list many of them, but out of Andrea Gore's lab at University of Texas, we've seen differences in physiological and sociosexual behaviors. And out of Emily Rissman's lab in North Carolina, modified expression. This is actually in the F3. That is actually a transgenerational rather than an intergenerational phenomenon. Some difference in brain and behaviors and social behaviors. With tobacco, we have seen some studies in humans, and if you attended the smoking workshop, you heard Dr. Svanes talk about some of these in some European cohorts. But we've seen risk for autism seems to be increased with a grandmaternal smoking. We had a presentation also by Gayun Yim that has not yet been published, showing an increased risk for ADHD in the grand offspring. Dr. Svanes and Dr. Holloway talked about increased risk for asthma and allergies. And in addition, it's not just epigenetics. As Dr. DeMarini can tell you, tobacco is likely a human germ cell mutagen. And as Dr. Yauk and Dr. Marchetti can tell you, most likely it has a widespread population-based impact on intellectual health. In mammals, we're also seeing worrying trends with increased risk for pulmonary dysfunction and also ADHD-like behaviors. Now, finally, onto the volatile anesthetic gases, which is really my focus at this time. Are we seeing anything there? Well, anesthetic gases really, it's not an exposure that I've really heard discussed at EMGS, but I think it really should be. I think it should be one of the exposures of top concern for this society, but I'll talk about it a little bit. They are small lipophilic molecules, and this is just the gases. There are also many other kinds of anesthesia that passively diffuse through cells and enter the nucleus. The agents work on many receptors. They suppress excitatory paths and augment inhibitory signals. Up to 75,000 pregnant women per year, and therefore fetuses, are exposed. 1.5 million infants out of 6 million children, and about 48 million surgical inpatient procedures per year. And what we do know is that GA exposure can impair germ cell quality. So that would mean things like morphology, concentration, motility of sperm. GA also disrupts normal reproductive hormones, so some of those abnormalities can be traced to that, apparently. It is also genotoxic. I don't think it's known as a mutagen per se, however. And genotoxic, that's in peripheral blood. And there's been almost no research on oocytes, but what has been seen is injury to oocytes and alterations in hormone levels. So what has been studied fairly well is the somatic toxicity of general anesthesia. It's known to cause learning impairments and behavioral abnormalities when administered very early in life, before the age of 3. Dr. Martinyuk, who's over there, talked about that in his presentation yesterday. The mechanisms involved seem to include neuronal apoptosis, impairments in synaptogenesis, neuronal migration, and disruptions in the GABAergic system. So GA is known to be a powerful modulator of chromatin remodeling with changes in key transcription factors, histone modifications, and that derailed transcription of genes that are important for neurodevelopment. Now, what's kind of intriguing to me is that in post-mortem studies of autism brains, of course this has nothing to do with GA per se, but when you look at those studies, they see those same phenomenon: failure of cortical neuromigration, failure of proper cytoskeletal formation, failures in synaptogenesis, neuron loss, and disruption of the GABAergic system. So it's just amazing to me how much these two areas overlap. Now, there's not been a single human study on the heritable effects of general anesthesia. Not one. These drugs have been around since the late 1950s. Not a single study. You can give a guy, 10 doses of general anesthesia, and it's never seemed to occur to anybody that might have a heritable impact on his potential offspring. But there have been a handful of mammal studies on this. The first two were in the 1980s, early 1980s, out of NYU. These were not mechanistic studies. These were just very, very small studies that, to I think what seemed to be the laboratory's surprise, found learning impairments in the progeny of the exposed germ cells. So these were older anesthetics, halothane and enflurane, but both when there was a fetal germ cell exposure and a later in life sire exposure to the sperm, they found what seemed to the researchers to be genetic alterations that affected brain development. Now, 34 years after these really, what should have been, in my opinion, very alarming observations, that should have triggered a cascade of, an avalanche, if you will, of papers. Instead, you have complete silence with nobody even thinking about heritable effects of GA until just last year, Dr. Martinyuk's lab at University of Florida. They published a paper in last year, and just very recently, just I think this month, in the Journal of Anesthesia. And I can't do it justice, but basically this lab did find that in the male offspring of the exposed germ cells, not the female offspring, there were learning and behavioral impairments as tested by kind of standard battery of rat tests. And they also linked that to defects in methylation. So should we be testing for, with all of that in mind, should we be testing for heritable impacts of germline toxicant exposures, of course, I'm here to say emphatically yes. If you want to know more about my perspective on this, I published this year in EMM on in a commentary that's specifically about fetal germline exposure, but of course all germline matters as well. And I think that not only should we be concerned about these exposures for the future, but I think we need to be concerned about the glut of exposures that happened in decades past, especially in the post-war eras when pregnant women, for example, were smoking heavily and were very heavily medicated. A pregnant woman in the 1960s had an average of 5 prescription drugs while pregnant. It's nothing like today. What are the legacy effects of those exposures? Well, it's hardly been studied. We don't know. We're seeing an increase of some very serious pathologies in children: asthma, allergies, autism, ADHD, and others, infertility. And really, we've barely thought about whether we can connect that to old germline exposures. And also, we do emphasize in this paper, because I wrote it with the head of DES action, that the generational impacts of DES is a vanguard question that deserves more study. So I just referenced the increase in autism, and I guess that's sort of an elephant in the room, because I think there's a popular conception that the increase in autism is an artifact of better diagnosis, changing definitions, greater awareness, blah, blah, blah. I'm here to tell you that is emphatically not true. We are seeing in our communities throughout our country and throughout the Western world a very dramatic and a very devastating increase in very serious neurodevelopmental disabilities in our children. And this is just a slide from the Department of Public Health in California. I didn't make up this slide. This is the prevalence. This isn't caseload. This is prevalence of developmental disability type autism. These are the autism cases that enter our developmental disability system in California from birth year 1990 to 2012. And we've seen a steady increase. This does not include the high functioning people. This does not include, the people who, who can advocate for themselves or who maybe have Asperger's or whatever. This is only a part. This is only the more severely affected part of the autism spectrum. This is comparing apples to apples over time. And in fact, in the California system, we've made it more difficult to enter the system over the years, not easier. We haven't brought in the definition. We've made the definition narrower. So if anything, the number should be going down. So we see now nationwide, and I do want to comment here just really quickly, 2% of all males born in California now end up in the developmental services system as autism cases. 2% of all boys. There is no precedent for this whatsoever, and it makes me absolutely insane to hear in the media over and over again that we're just noticing it better. This system would have noticed this. These are not cases that you miss. Now, in nationwide, we're seeing a trend towards higher than 1 in 59. Now the epidemiologists think it's more like 1 in 40 of all children born in the US ending up having autism. But strangely, this is a highly heritable disorder. We know that. It's a highly heritable disorder that's been increasing in prevalence and is not strongly genetic. There is a great deal of missing heritability here. But strangely, we really haven't thought about germline apart from the DNA. And almost all of the research into heritability of autism, I'd say 99.999%, is in nucleotide sequence. There's almost no effort looking into the exposure context. Why is that? How come no one's thinking about our germ cells? There's no seat belts for our germ cells, it feels like. Well, at the FDA level, and believe me, I've petitioned the FDA to look at fetal germ cell exposures, and I've been rejected twice very solidly. They really don't give consideration to germline or heritable effects of pregnancy drugs or other drugs. As I said, I've had 2 citizens' petitions denied. I'm proud of it. The Center for Tobacco Products at the FDA, we couldn't even get them to come to our Heritable Hazards of Smoking workshop. It's not, germ cells are not even on their radar at all. At the EPA, we have Dr. DiMarini, who has been successful in introducing mutagenesis as an endpoint of concern, or germline mutagenesis as an endpoint of concern. But from what I can tell, there's not really a whole lot of effort there to look beyond, like an Ames assay. I could be wrong about that. type of test.
You're not wrong.
I'm not wrong, says Dr. Zumarini. And then from the research side, instead of the regulatory side, the NICHD, from what I can tell, they do some research on basic germ cell science, but they're really not concerned about germ cell exposures at all yet. The NIMH, very, very concerned about autism, ADHD, mental health, mental illness, genetics, not at all concerned about whether there could be other germline consequences. NIEHS, I think they are far ahead of everybody else in the country, and they're mostly concerned with environmental exposures and not so much these higher dose toxicants like these pharmaceuticals and smoking, but that's because that's their purview. I have made nominations to NTP and OHAT to start looking at these things, however. And for the CDC, like, forget, not even on their radar. Like, you talk to the CDC people and they think you have like horns coming out of your head and 3 eyes and purple hair. They think you're crazy. So in summary, I'm here to say that I think that there are 3 routes to pathogenesis, and we've only been focusing on 2, which is inherited genetics and environmental somatic, but there's this third route, the third leg of the stool, if you will, of pathogenesis, which is germline exposure, which unfortunately has been pretty much ignored. And that these germline exposures to toxicants can deregulate gene expression during the offspring's development or even result in mutagenesis as well. And if we want to understand the roots of heritable but non-genetic disorders such as autism and ADHD, we should consider germ cell exposures in addition to genetics. And that's all. I obviously have no lab to thank, but I do want to thank the EMGS and Dr. Holland for having me, and I'll be happy to take any questions if there's time.